Yeah, so big thank you to Marie and Gabe for organizing this whole workshop and also for inviting me. I'm really excited to talk about some of my graduate work today. So as Maria mentioned, my name is Vicky. I'm a grad student at UChicago in Sally Horn-Bedevinek's lab. And I will move my cursor out of the way. There we go. And so our lab studies collective cell migration as well as the basement membrane extracellular matrix, which is what I'll talk about today. And so I am a developmental biologist. Sorry, oh, yeah, there we go. I'm a developmental biologist. I'm really interested in how organs acquire their distinctive forms. So I'm showing you guys the lungs and the mammary gland in the mouse, as well as the lymph organ in Drosophila, all of which have wildly different forms that help them achieve their specific functions. And what I'm especially interested in is how constraint from the extracellular matrix can shape an organ. So what is an ECM for my non-biologists? It is a complex network of proteins and glycans, aka carbohydrates, that is on the outside of cells and helps support their functions. So some ECMs that we might be familiar with are bones, teeth, seashells, or for my skincare enthusiasts, you've heard about like collagen making your skin elastic. That's all ECM. And so ECMs have a ton of functions within our bodies. What I'm most interested in, as I mentioned, is how ECMs can hold tissues together and also constrain growth. Obviously, they can't squeeze themselves, but they can act as kind of like a wall for cells to butt up against. And that is really how ECMs contribute to morphogenesis. And then, of course, they also have a bunch of functions in physiology, cell signaling, and protecting tissues from the environment. So we've all got them. They're super important. And the specific ECM that our lab studies is the basement membrane, which is this very thin, compact, sheet-like ECM that lines our epithelial and endothelial tissues. And so if we look at this little zoomed-in diagram of what this ECM actually looks like, we'll see it's made of these interconnected networks of collagen 4 and laminin held together by perlecan and nitrogen. And a great thing about the basement membrane is that no matter what organism or what tissue you're looking at, this core structure is going to be remarkably well conserved. So as we'll see in a second, I work on fruit flies, but they have the same core structure as our own basement membrane. But one issue that we do run into, especially if we're interested in morphogenesis, is that if we look at all these examples of basement membranes in our body, so for example, in our capillaries or in muscle, you might notice that they're involved in pretty complex morphologies, and oftentimes they're also embedded pretty deep within tissue layers. And so in a lot of these systems, we lack tools to manipulate or even to visualize an ECM in its native environment. And so to address some of these challenges, our lab, like I mentioned, works on Drosophila, aka fruit flies, and specifically on an organ-like structure called the egg chamber, which is what will go on to form Drosophila eggs. So if we look inside the ovaries of a female fly, I'm going to show you here a developmental array of egg chambers, starting with the youngest down here. And then you'll notice that as they get older, they both continue to grow in size and then also elongate, forming this ellipsoid shape in a more mature egg chamber. So now if we zoom in to each individual egg chamber, I'm going to emphasize again that our important shape transition here is going to be going from this sphere early on in development to this ellipsoid or kind of like football structure as they mature. And so if we look inside each egg chamber, we'll see that they consist of the oocyte in pink. This is what is going to form the egg. These cells called nurse cells, which feed the oocyte as it grows. And these are all going to be surrounded by another layer of epithelial cells called the follicle cells, shown in blue. And then the follicle cells will secrete that basement membrane extracellular matrix that I'm interested in, shown here in green, which is on the outside of the whole tissue. So just a couple things to note here before I move on. I'm always going to show you egg chambers oriented like this, with the oocyte facing towards your right. And then you might think that the follicle cells at the anterior here are gone, but I promise they are still there. They're just very thin and stretched out. So I mentioned that the follicle cells make their own basement membrane, and the way that they do so is coupled with a process of collective migration. So I'll show you here the follicle cells shown in pink. You'll notice that they are migrating as a collective, and they do so with the basement membrane shown here in blue as their substrate. And so as the follicle cells are migrating, they're also secreting and remodeling that basement membrane that they're crawling on. So again, here the follicle cells will be in pink, and then in white is fluorescent collagen. So this is that matrix. And you might notice that those little bits of collagen are being secreted and kind of like pulled down by the cells as they go, marked in those colorful lines right there. And so this creates a pretty specific mechanical pattern in the basement membrane, a stiffness gradient with much softer basement membrane out at the anterior posterior poles, but stiffer in the middle. And so if you now imagine that tissue on the inside wants to expand out relatively evenly, but it's experiencing a lot more constraint from that stiff basement membrane in the central regions. And so it's going to follow the path of least resistance and go out towards the poles. And that is how we get elongation of this tissue. This is what the field refers to as the molecular corset model, because you're getting squished in the middle. And so really, this is our preeminent way of thinking about basement membrane mechanics in this system, but there is a problem, which is that I showed you that this molecular corset is built up through this process of cell migration and remodeling of the matrix, but that migration only persists for the earlier part of development. So at a certain point, that migration stops. We've lost out on the mechanism that builds that corset, and yet the egg chamber needs to both continue to grow in volume and also keep elongating along its AP axis, even though we've lost out on the mechanism we think is responsible for that elongation. So this is really what my project kind of originated out of, is this question of how are we still getting elongation? And the field has just assumed that, okay, whatever corset was present in those early stages is going to be sufficient to continue controlling elongation for the rest of development, but we actually now know that that is not the case. So the first thing that we know is that the mechanical pattern actually changes really dramatically, going from that symmetrical stiffness gradient I showed you during migration stages to then having this really distinct half and half distribution of a soft anterior basement membrane, but a stiffer posterior. And then at the same time, there's a big change in the growth of the cells on the inside of this tissue, going from largely uniform earlier in development to then at these post-migration stages, we're getting closer and closer to becoming a mature egg. And so that oocyte here at the posterior is getting really large. And so overall, what I think is going on at this point is that that growth of the oocyte at the posterior is being constrained by that stiffer basement membrane, but all of that tissue growth still needs somewhere to go. And it's going to get pushed out towards the anterior, where it might be enabled to do so by a softer, squishier, more deformable basement membrane. And so when I first embarked on this project, kind of the first order of business was to look at the composition of the basement membrane at these stages, because broadly we know that what's in a matrix can affect its mechanics. And so again, I want to show you these core structural components, collagen 4, perlecan, and laminin. And we'll see that at those migration stages, their distribution is largely consistent. When I first looked at collagen, I'm like, this distribution looks the same. This wasn't really that surprising to us because we already know that collagen is not being made between these stages. And so whatever network was there just gets stretched out as the tissue gets bigger. But when I looked at perlecan, which we usually associate with a softer basement membrane, I found that it is strikingly enriched in that same anterior region where we know that the basement membrane is softer. And conversely, when I looked at laminin, which again is this key structural component, it seems to be lost from that same anterior region. Again, softness. And so as I'll explore in the rest of this talk, I think this is a really unique opportunity where we have a very tractable system. For anyone that doesn't work in Drosophila, we have amazing genetic tools that makes this kind of unique in terms of we can actually manipulate and visualize the basement membrane, which is not something that is available in all other systems. And we also have this really interesting change in composition and mechanics, where again, there's this half and half distribution of soft versus stiff. And also we know that the composition of the matrix is very distinct across these two different regions. And so I think this is a really unique opportunity for someone like me who's interested in ECM mechanics, and especially in a developmental system, in a living organ. We actually have a system where we can interrogate the basic material properties of the basement membrane, which I think is very exciting. So in this talk, I'm going to first talk about how the collagen 4 matrix stretches on a developmental timescale. And then going to ask what happens if we expose the matrix to more acute stress. And then kind of going off of that, can the basement membrane recover from stress? Might that be different in these two very different regions of the matrix? And then finally, in the spirit of this workshop being about collaboration, I want to highlight a collaboration that we just started with another lab at UChicago, which hopes to kind of bring everything together and ask how the material properties of this basement membrane might ultimately inform tissue elongation. So first, looking at collagen 4 stretching on a developmental timescale. I mentioned this earlier, but collagen 4 is not produced from going to these migration stages to post-migration. And this is kind of unique in the basement membrane field because collagen 4 is considered kind of our main mechanical contributor. And so it's kind of surprising that the tissue still needs to grow so much, and yet we're not making this like really important component of the matrix. And so there was this question of, presumably, this has to be stretching to accommodate this big growth in volume, but we don't really have a way to confirm that. It's just kind of intuitive. But so one of my ways to interrogate this was using a fun genetic trick that I can do in flies, where I basically heat shock the female fly and make these single cells that are making a fluorescent collagen. And so you'll remember I showed you earlier that those cells like to migrate as a collective, and they're also depositing matrix as they do so. So now those little single cells are going to deposit these fluorescent stripes of collagen that are going to remain in the matrix and make these little stripes that are about 1 cell width wide. So now if we then leave these egg chambers still within the female fly to develop normally, and then take them out and look at them at the post-migration stages that I'm interested in, we can use the width of those stripes as a proxy for how much that region of the matrix stretched out over the course of development. So I'm showing you here what I thought I would see with this assay, where again, I think that that anterior region, so this part right here, where the matrix is much softer, I think is going to be a lot stretchier. And so I would expect the stripes to be a lot wider. So here's what these look like in an actual egg chamber. At stage 8, this is right when migration ends, and then also stage 10, this is post-migration. And if we actually zoom into these stripes, we'll see that they start off about 10 microns or 1 cell width wide, and those little bits of fluorescent collagen are going to be really close together. But then if we look at them at those post-migration stages, not only is that stripe now about twice as wide, but the little bits of GFP-tagged collagen are spaced much further apart, confirming that, indeed, the matrix is actually stretching over the course of development. And I mentioned earlier being interested in whether there might be a difference between how much different parts of the matrix might stretch. And so I quantified the width of these stripes, first at migration stages. So we see that they start off about the same width in both the anterior and the posterior. But when we look at post-migration stages, as I predicted, the stripes in the anterior region, so again, that softer basement membrane, are wider, indicating more stretching on a developmental timescale. So this is great. This kind of agrees with my hypothesis. But I then wanted to ask, what happens if we have a little bit more of a dramatic condition where we expose these egg chambers to much more acute stress? So to do this, I used what's called an osmotic swelling assay, where I take the egg chambers and place them in a hypotonic solution. This is going to cause water to come into the tissue and everything is going to swell. And I can combine this with photobleaching fiducial marks into the fluorescent basement membrane, which should also increase in size as the tissue expands. And so again, I'm showing you here what I thought I would see with this assay based on what I just showed you with the stripes. We think that the anterior region is stretchier, and so those marks will be bigger in the anterior. So here's what these marks look like, photobleached into, in this case, fluorescent perlecan. And then what we see once we expose them to this osmotic swelling, over the course of about 20 minutes, you're going to see water come into the tissue. It starts to expand. Those marks will also start to increase in area. But what I started to notice, if we just pause at this last time point, come back. After about 20 minutes of this osmotic stress, I've overlaid kind of the original sizes of those marks onto that 20-minute stress time point. And what I started to notice is that, contrary to what I expected, the marks in the posterior region, that stiffer matrix, looked bigger, which was kind of confusing to me. And this is a trend I saw really consistently. So I'm showing you here my control where there's no osmotic stress, as well as this osmotic stress condition. Each of these little dots is going to be the average of marks for one egg chamber in their respective regions. And this is like a really consistent trend that I would see where the stiff posterior is deforming more. I wanted to interrogate this a little bit further and see if maybe this is just an issue with my assay. So I tested a couple different osmotic stress conditions, so both a milder condition and also a more high stress one. And as you might expect, the overall amount of the tissue deformation expands with how much stress you're putting on it. That's kind of intuitive. But this trend of the stiff posterior expanding more was really consistent. And so with these two different assays I've showed you, obviously they're very different magnitudes and timescales of stress, but the basement membrane seems to be reacting in different ways. So with these collagen 4 fluorescent stripes, we have a developmental timescale of about 30 hours. And in this case, we see greater stretching of that soft anterior basement membrane region. But with our osmotic swelling assays, we have very rapid tissue expansion. Obviously, this is a non-physiological condition. And in this case, we see greater deformation of that stiff posterior. So I'm thinking about this right now is that maybe on a developmental timescale, that stiff basement membrane is really good at constraining tissue growth. But when you plop it in this osmotic swelling assay condition, it's like, we can no longer resist expansion. We're almost like brittle, despite being stiff. So this is obviously very different results from these two different assays. And so I wanted to take this osmotic swelling assay one step further and ask, what happens if I remove stress from the basement membrane? Can it recover? And again, is there a difference between these two different regions of the matrix? So how I would do this, they've been hanging out in this hypotonic solution and swelling. I'm going to now return them to an isotonic solution and remove that osmotic stress. And there's two different responses we might expect for those little fiducial marks. One being a viscoelastic response, where there's some deformation of the tissue, but when the stress is removed, everything can kind of come back to its original shape. Or we might see a more viscoplastic response, where again, there's deformation of the tissue. And once the stress is removed, it can come back a little bit, but not return to its original form. There's been some sort of irreversible or plastic deformation. So what happens to our egg chambers? I'm going to show you 5 minutes of that osmotic stress being applied. Already we can see that that posterior is deforming more. And then once the osmotic stress is removed, what we can see is that the soft anterior can return back to its original form, but the stiff posterior cannot. There's been some sort of irreversible damage to that matrix. And so I'm really interested in how composition might play into how these different parts of the matrix react to stress, because I'll remind you again, we have this very distinct compositional pattern in our wild type or normal egg chamber, in particular that we have a lot of this soft protein perlecan in that anterior region. And so I think that that could potentially be part of what is responsible for its ability to return back to its original form after stress. And so again, we have a lot of genetic tools that we can make use of in flies, which is great for me. I can knock down perlecan in only the anterior region and ask, now what happens? Does it behave more similarly to that maybe stiff, brittle posterior? And so I'm showing you here 10 minutes of that osmotic stress being applied. And what we see once that stress is removed is that when perlecan is removed from the anterior region, it can now no longer return entirely to its original form, suggesting that actually having perlecan in that matrix might be contributing some elasticity to it. And so this relationship between composition and mechanics is something I'm really interested in and is what I'm working on at the moment. So stay tuned and see if I can figure out what each of these components do. So I've shown you that the anterior and posterior respond differently to these different forms of stress and also recover from stress differently. And so in the last couple minutes, I want to highlight this very new collaboration that we have with Noah Mitchell's lab at UChicago, which hopes to bring all this together. I mentioned that I'm interested in using the egg chamber as a really tractable system to study fundamental physical properties of the basement membrane. And we want to bring that together and figure out how it corresponds with egg chamber elongation. So as I mentioned, this is being done in collaboration with Noah Mitchell at UChicago. And most of the work is going to be done by Sam, who's an undergrad in the lab, under the guidance of Nico, who's a postdoc in the Center for Living Systems. And so what Sam is working on at the moment is taking egg chambers at all these different developmental stages and developing an algorithm to basically get the radius at all different points along the AP axis for each stage. And from that, he can get a cylindrical parameterization of egg chambers by developmental stage. So from there, the Mitchell lab has this new simulation tool that we want to use to infer the stress on the basement membrane at different points based on tissue geometry at all these different developmental stages. I want to combine that with my experimental data on the basement membrane's response to acute stress. So this is going to give us an idea of the basement membrane's in-plane deformation when exposed to stress. And also combine this with data from the literature, primarily atomic force microscopy data, which gives us some stiffness measurements, not in plane, but if you poke the basement membrane like that. And we hope to bring all of this together and develop a model of egg chamber morphogenesis throughout development based on both the inference of stress on the basement membrane, based on the tissue's volume and shape changes, and also what I've now found about the material properties of the matrix. So to recap, I've showed you on a developmental timescale that soft anterior matrix stretches a lot more. Under more acute stress, it switches. Now the posterior might be a little bit more brittle and get deformed. These two different regions also have different responses once you remove stress. And I'm currently investigating how composition might play into all that. And with our new collaboration, hopefully, fingers crossed, we can put all this together and figure out how these material properties ultimately will inform tissue elongation. So with that, I want to say a big thank you to my lab and my thesis committee, the members of Noah's lab, and the funding that makes this work possible. And I can take questions.
Super cool. I'm wondering if you, okay, as a theorist who likes to build minimal models, if you want to be like really naive and of course, like obviously your nose group is going to do this in a very sophisticated way, I'm sure. But like, if you wanted to be really naive and just say, my intuition tells me that if this is something which is like elastic and viscous and maybe plastic in some way, and I wanted to assign like which like what the composition, what the perlecan and what collagen does, so like all three of those parameters, like what do you think like they build into?
Yeah, I think definitely. So I'm not the first person to suggest that perlecan equals elastic. So biochemically, we don't super know how this happens, but there seems to be this inverse relationship with collagen being our like contributor of stiffness. And I guess it wouldn't be elastic because I think you stretched it, it wouldn't come back. It's like very much you stretch it and it breaks. And then perlequin in theory is what is contributing that ability to kind of come back. So I guess that would be the elastic component.
It's like in principle, like in some sense, maybe perlequin is like it's like stretchier as a thing itself, right? And then you can
Yeah.
go back versus something that you tear when you stretch it too much.
Yeah, there's one component that's really interesting that I have no idea how to experimentally test is that perlequin as a molecule loves to hold on to water. And so my way I'm imagining this is that that anterior region of the matrix that has a lot of perlequin in it, I think is like very hydrated and squishy. And I would love to like have ways to mess with that. I have not been able to find a way. And it's also like the basement membrane is so thin that I don't know how I could even visualize it, but that's also something that's going on. So
I might have missed this. In your experiments with the osmotic changes, how do you decouple the effect on the ECM and the effect on the cells?
yeah, so I'm not the first person to use these osmotic swelling assays. They're definitely not perfect because, as you're mentioning, I think there's going to be differences in even how different types of cells swell. Overall, the ECM is considered to be so much stiffer and more robust than the cells inside that everyone who uses these swelling assays is like, any changes that are happening, like the ECM maybe taking up more water is not necessarily going to play into this. And it's also so dramatic that the cells are like, we're expanding. And so there's any minutia of like how much the matrix itself is taking in water versus how much the cells are swelling, we're negating. And then for the cells on the inside, at first I was really worried that like, obviously this oocyte here is very different from these rest of these cells. Maybe they're swelling differently and my assay is just bad. And so if I digest away the basement membrane with a collagenase and then just let the cells be exposed to osmotic stress and swell, they swell relatively equally. Again, if you, I don't know, maybe looked at this with a much finer comb, maybe they're not exactly equal, but for this kind of brute force, we're treating it as everything is expanding out. Yeah, Sid.
Sort of as a follow-up to that question, do you think, or in previous swelling assays that have been done, do you think there's a chance that the extracellular matrix can play a role in the rate of osmotic change between the surrounding fluid and the cells?
Yeah, potentially. So yeah, like I mentioned, perlequin likes to hold on to water. Maybe that affects the rate at which water is able to kind of get through. Again, the field considers this such a kind of dramatic and obviously non-physiological assay that everyone is just like, the basement membrane itself is not that important because so much water is coming into the tissue that everything is just going to expand outwards. But definitely like within the female fly, I totally buy that the different parts of the matrix make them more permeable.
So it's like in the two parts that you have, it might be that the part that you expect to swell more, but it is not, might be because it is not able to get in as much water as it wants to, but it has capacity to take more water.
Maybe. Yeah, yeah, that would kind of, it would be an issue for my assay, so I'm hoping that's not the case. But yeah, obviously the different parts of the matrix are very different, so there's always a chance that what I'm measuring is due to some other extraneous factors.
I think I'm just trying to think about spinal cord injury in this case because, again, F actin is something that axons try to regrow within spinal cord injury. And one of the challenges is, again, how these axons are going through this inflamed extracellular matrix within the spinal cord. So kind of applying these concepts to kind of see and look into translational aspects of how spinal cord injury is going would be very exciting too. So I think it's very similar, even proteins and some of the challenges with the extracellular matrix. So very cool.
Yeah, thank you.
You did the collagen 4 assay where you tried to quantify the changes in collagen. Have you quantified something similar with the perlequin? Are you like, do you see like statistical differences?
Yeah, I would love to do that. Unfortunately, the way the genetics works out, it's a pretty complicated, like you have to cross a million flies to each other. And so using it with collagen is kind of the only way that we've been able to do it so far. And then also because perlequin is being made still at these post migration stages, you might get some complications between like, it's not necessarily that the stripe was left over, got stretched out, but like maybe you're now glomming on more perlequin and so it would get kind of messy. But I do think it would be really interesting to see like, this is how much the collagen 4 matrix is stretching. And like all these parts are interconnected. So my base assumption is that the collagen 4 stripes are reflective of the matrix in general, but there could very well be a difference between how much different components are moving around.
Do you see any different, have you done like omics on this? Do you see any difference in transcription? Okay.
Yeah, I have definitely not done any omics. It's very much not my field. I'm trying to think if anyone else in the field has done, I think there's been some single cell sequencing stuff that's on like our fly base. But the only thing that comes to mind is that these cells in the anterior are pumping out a whole bunch of new perlequin, which is something I've been able to confirm through RNA fish. But other than that, I don't know.
In terms of why the different mechanical properties at the different ends of the egg chamber matter, like what do you think is the most important impact of them on development?
Yeah,
not shocking the cells after.
I think in general, so I think laminin is going to be our #1 most important just because of the order of matrix assembly. Laminin always comes first. I didn't really talk about it today, just because it's harder for me to like experimentally manipulate. But basically, if you don't get laminin laid down, none of the rest of the matrix can come. So I think laminin is going to be always our most important one. And then after that, I think collagen, as I mentioned, is what we consider like our main contributor to mechanics. And so for translational people, I think most like basement membrane pathologies are going to be collagen mutations. And then I'm very interested in perlequin, as you guys can see, but I don't think we know that much about it in other systems necessarily.
Is their primary role like maintaining the shape or maintaining the mechanical stability or some kind of like pressure-based signaling to the cells on the inside? Do you have a sense of like
what they're doing?
Yeah.
Let me think. I think in general, it's going to be about kind of maintaining, almost like think of it like a scaffold for the cells. So it's like providing some kind of like mechanical stability in terms of we have stuff to build on, and then also keeping like shape. So I like to think about like, for example, blood vessels, like they have to have enough mechanical integrity to resist blood flow, but also some flexibility so they can kind of go like this. In terms of individual components, if I knew more about signaling, then I could answer that better because I think there is going to be some interesting how much different components will influence like morphogens and other cell signaling stuff, but off the top of my head, I don't know.
Sorry, this is super cool. Thank you. Do you see any evidence for like you showed in red and you circled our attention to like the boundary conditions having some anisotropy of like stiffness here, different stiffness there, and like then also the bulk. But if you like, do you have any anisotropy? There we go. In the bulk material properties, like if I tug on one end in a directional way, like some oxidic materials would like squeeze versus expand based on the Poisson ratio.
No idea. Yeah, I think a lot of these are experiments I would love to do, but it's like these guys are so small. So they're like 500 microns, and so even the AFM I think is kind of the limit of like what we can physically do to them, and then these swelling assays. But yeah, I would love to like even decellularize the basement membrane and just have the matrix on like one of those cell stretchers, but it's so small that I have not tried to embark on that. I think it would be wonky. Yeah.
Thank you.
Thank you so much.